Hi, welcome back to this video. So in the last lecture we saw that in prokaryotic populations, diversity is generated via novel mutations that are occurring in the population. Mutation rates are small, but yet the diversity that is generated in microbial populations is huge because of the large population sizes. We saw in something as small as just 100 milliliters of a flask in a lab, the population sizes can reach 10 to the power 11, and the number of mutations that can happen is of the order of 10 to the power 8, which is 100 million different mutations that happened in just that flask. And as a result of this, the variation that natural selection acts on is generated via these mutations, which is, and what we want to understand today is that if we are talking of eukaryotic and sexually reproducing organisms such as us, the diversity that is generated in the populations arise from a different mechanism altogether. I mean, mutations are the ultimate source, but if we are looking at some generations, the source of diversity is something else, and that we want to investigate. As a result of that, because the mechanisms via which diversity is generating in prokaryotes and eukaryotes are different, the approach to studying evolution for these two organisms is also different. In prokaryotes, which is what we will be studying through this course, we will be actually able to follow what mutation arose, at what time did it arise, what happened to it, what did it do, and understand the molecular underpinnings of how adaptation was facilitated. In eukaryotes, the study of evolution of eukaryotic sexually reproducing complex organisms takes the name of population genetics, which is something that we will not be talking about this course. But it's important to understand that distinguishing feature between the two, and this short video is sort of an attempt to understand what happens in sexually reproducing complex organisms. So, sexually reproducing complex organisms such as us why are we saying that, like in the case of prokaryotes where new mutations are happening and that is leading to generation of variation, why is that not the case here? So, the first thing that we must recognize is that inheritance is biparental. So, you have parent 1, you have parent 2, and these two mate to produce an offspring. This is qualitatively different from there being one parent which divides into two offsprings. Now, the DNA of this organism is coming, half the DNA, 50% DNA is coming from one parent, 50% DNA is coming from the other parent. This is qualitatively different from what we have been looking at so far. And before we go any further, we first need to look at how is our DNA organized inside the nucleus. By our DNA, I mean any sexually reproducing complex organisms, but human beings are a good system to sort of look at this problem. So, inside each of my cells, let us say this is a cell, our cells are much larger than bacteria, is the nucleus. Most of our DNA is present in the nucleus. There is some in mitochondria, but that is minuscule and we will not discuss that in this context. Unlike bacteria whose DNA is circular in nature, human DNA is linear threads of DNA, and there are the linear threads are called chromosomes. So, there is chromosome 1, chromosome 2, 3, going all the way to 22. They get increasingly smaller. The numbering is because of the size. And the 23rd, I am a male, so I have X and a Y. Well, let's discuss the 22 first, and we'll come to the last one later on. The interesting part is, because inheritance is biparental, I don't have one copy of chromosome 1, I actually have two copies of chromosome 1. So, the other copy that I am carrying, I inherit one copy from each of my parents. So, I have two copies of chromosome 1, one from my mother, one from my father. I have two copies of chromosome 2, I have two copies of chromosome 3, and I have two copies of chromosome 22. In addition, the 23rd chromosome is what determines the sex of the individual, and in my case, since I am a male, I inherited the X chromosome from my mother and the Y chromosome from my father. Y is much smaller than X, and this forms the 23rd pair of the chromosome. So, human DNA is organized in these 23 pairs of different threads. Each one is a double helical DNA structure, and this inheritance is biparental, because the blue colored DNA is something that I have inherited from my father, and the other color is from my mother. So, then the question arises that what do I as an individual whose DNA is this pass to my offspring? What is the 50% that I pass through? So, obviously, I pass it through the sperm, and I am going to pass one copy of each of the chromosomes. When I say that one copy of each of the chromosomes for chromosome number 1 going all the way to chromosome number 22, I will pass that. In addition, I will pass either X or Y for chromosome number 23. So, the question then becomes that since I have two copies of chromosome 1, this and this, which of the two copies do I pass to my progeny? Is the choice completely random? Does one have a greater chance of being passed on over the other? And that is the question that we will be looking at, and that is the question that makes sort of sexual reproduction quite amazing. And this 23, there is nothing sacrosanct about 23, it is just that we happen to have 23 pairs of chromosomes. Different species have different numbers. Something like Baker's yeast has, which is a single celled eukaryote but capable of sexual reproduction, has 16. You will find examples of different numbers throughout nature. So, we will only look at, let us only look at chromosome 1 and see which of the two copies gets passed to my progeny. And the chromosome 1 that I have, the two copies of chromosome 1 that I have are arranged like this. Oops, sorry. One copy of chromosome 1 and the second copy of chromosome 1, and we will look at chromosome 1. So, this is one copy from one of my parents, this is the second copy from my other parent. And the question is, which of the two do I pass to my offspring? And what nature does here is something very clever and quite complex. It's So, the copy that's passed to the progeny is neither this one nor this. What nature does is that it shuffles the two chromosomes back and forth and creates a hybrid, one copy of hybrid chromosome. What it might do is that it will start with this one, and then at some point in the chromosome it jumps to the other copy, and then it continues with this one for a while, and then it switches back to the original, and then it may copy this for a while, and then it may switch back to the second chromosome again and go all the way. If this is how the chromosome 1 was packaged, then the hybrid that you would get would be something like, the blues would be separated and in between them would be this orange colored strand, and this would be the hybrid chromosome. This process of switching back and forth is called recombination. And for every chromosome this happens, and you have a hybrid one which gets passed on to the progeny. These switches happen at least 3 or 4 times on every chromosome. On an average they happen every 3 or 4 times and create this. Their exact locations are, again, these are statistical properties. Their exact locations are, there are no exact locations, but statistically we know that these regions have higher chance of crossover from one to the other than other regions in the chromosome. Because this process is not certain, the chromosome 2 might look completely different. Chromosome 2 might look something like this. Chromosome 2 is smaller than chromosome 1, but this is what it might look like. So, you can see, so this is chromosome 1, this is chromosome 2. And as you can see that in chromosome 1 it is relatively 50-50, in chromosome 2 maybe the blue is a little more, and you can have all sorts of variation. Now, this process of shuffling these pairs of chromosome and generating this hybrid chromosomes, one copy of chromosome for each one of the 22, and then packing one of X or Y, would be done by me. The same would also be done by my partner, and then that is how the fusion of these two would lead to birth of a creation of a offspring. Now, if this process of shuffling and recombination matters, because imagine the scenario where the two copies that I am carrying, again I got one from my father and one from my mother. Let us say we are talking of chromosome 1. Imagine the two copies that I am carrying are identical to each other, are completely identical to each other, which means the DNA sequence of this, let us say this came from my father, let us say this came from my mother. If these two sequences are completely identical at every single place, which means the Hamming distance between these two sequences is 0, then recombination does not really matter, because you can go up to this point and then switch and then copy from here. But because copying from here is actually exactly the same as copying from here, it does not really matter which of the two chromosomes you copy from and create this hybrid, because both are identical at every single place. So, it does not really matter. However, chromosomes are not identical. Even though between any two human beings there is large amount of similarity in the DNA, there are enough differences for something like this to happen. Let us imagine that they are not identical, in fact they are carrying these mutations. If I identify the locations of the differences in the DNA sequence between the two copies of chromosome 1 that I am carrying, let's imagine that one is represented by a plain line and the other one, every time there is a difference between the two chromosomes, I identify that with a red line on the second chromosome. And now you can see that the number of hybrids that I can create is really high. Now as the process of generation of this hybrid starts, I start off from here, but it matters now whether I am copying from this or this template, because there are these mutations here. So if I am copying from here up until this point, what that means is that I have missed out on these 6 mutations, and then I switch, and now I am copying this, but then I am missing on the variation that is here, because these two are non-identical. So the number of variations that you can generate via this process is astronomical, and remember that this is happening on one chromosome, but this is also happening at every other chromosome except the 23rd, because X and Y there is no recombination. So the number of genetic variations that you can create just by process of recombination is astronomical. As a result of that, the variation that is generated in complex sexually reproducing organisms such as us is largely through recombination. Of course, mutations are also happening, newer mutations are coming up all the time, but their contribution to the variation that exists in a population is relatively small as compared to the variation that already existed, and then recombination just creates different permutations and combinations of that entire process. This variation that exists in a population, so let me just redraw these chromosomes again. Let us say this, second one is the same color, but let us say the mutations are, these are the places where mutations. That is not to say that only this copy of the chromosome has all the mutations. These red marks are to identify locations where the two strands are not identical to each other. So the mutation could have happened anywhere, the result being that the two are non-identical, and that is what these marks identify. So the variation that is the variation that is generated, the key take home from here is that the variation that is generated is largely through recombination. Recombination and not via mutations. Of course, what recombination is doing is that it is creating newer genotype it is creating newer genotypes out of existing mutations. In the population pool that exist. Remember that if the two copies of the DNA were exactly identical to each other, then recombination does not generate diversity, then mutation is the only source of diversity. Recombination is able to generate newer and newer combinations of mutations because there exists considerable variation as it is in the population. This existing variation, existing mutation, and that leading to recombination and generating newer and newer variation, this existing variation in the population is called standing genetic variation. And complex sexually reproducing organisms rely on standing genetic variation in creating further diversity, genotypic and phenotypic diversity, and respond to the environmental challenges that they face. This is also important because the generation time of complex sexual reproducing organisms such as us is really large. So, transitioning from one generation, suppose an offspring is born today in a human population, you have to wait till 25-30 years before it leads to the next generation. So, the waiting time of generations is really large, and hence you need newer variations, newer mechanisms to create variation. Simply waiting for new mutations to occur and then test those out by natural selection as to which one increase fitness may not be the best strategies, and hence recombination is a great strategy which speeds up the ability of a population in terms of its adaptive speed in how it responds to environmental challenges. The point being that there are two different ways of looking at evolution of a population, depending on whether you are looking at asexual populations where evolution happens via novel variation, newer mutations that happens, versus if you are looking at sexually reproducing organisms where we are looking at standing genetic variation or the variation that already exists in the population. And for the purpose of this course, we will stick with prokaryotes, but it is important to understand and distinguish these two scenarios, and we will not come back to sexually reproducing complex organisms. So, we will continue our discussion of prokaryotes and how they evolve in the next video onwards.